So I would like to invite our next participant, Rishika Sharma. Rishika Sharma, are you there?
Yes, sir.
Okay, start with your presentation.
Yes, sir. So, good afternoon, everyone. My name is Rishika, and I'm currently pursuing BTech Biotechnology, fourth SEM from Maharishi Markandeshwar deemed to be University, Mulana Ambala. Uh, and my topic for presentation is role of nanoparticles as nanocarriers in drug delivery system. And I have prepared this PPT with some review articles and under the supervision of Dr. Manoj Singh. So, what are nanoparticles? Nanoparticles are the tiny particles with dimensions typically less than 100 nanometers. These materials exhibit unique properties due to their small size, like colloidal properties and ultra-fast optical effects or electrical properties. And they cannot be seen with ordinary optical microscopes, and they require the use of electron microscopes or microscopes with laser. Next, we have properties of nanoparticles. Uh, they have increased surface area, they have higher surface area to volume ratio, which impact the reactivity and interaction with the other substances. They exhibit size dependent optical properties influencing their color, absorption, and emission characteristics. Their size allows for increased reactivity, making them useful in catalysis and chemical reactions. Some nanoparticles, like certain metal and carbon based nanoparticles, can display enhanced electrical conductivity, and they are useful in the drug delivery. Here are some of the examples which are used in the drug delivery, like liposomes, polymeric nanoparticles, myceles, dendrimers, nanocapsules, nanospheres, etc. So why nanoparticles are used in drug delivery? They uh, Nanoparticles can be designed to target specific cells or tissues, improving the precision of drug delivery and reducing side effects of healthy tissues. Their higher surface area allows for efficient attachment of drug molecules, enhancing the therapeutic payload that can be delivered. Some drugs have limited solubility, and nanoparticles can help improve the solubility, making them more bioavailable and effective. They have a prolonged circulation time. They can extend the time a drug stays in the bloodstream, improving its circulation and allowing for a sustained therapeutic effect. Next, I have chosen the mechanism of drug delivery in brain. So here, nanoparticles for the drug delivery to the brain is the method for transporting drug molecules across the blood-brain barrier using nanoparticles. These drugs cross the blood-brain barrier and deliver pharmaceuticals to the brain for therapeutic treatment of neurological disorders. These disorders include Parkinson disease, Alzheimer disease, depression, and brain tumors. Some nanoparticles, like lipid-based cationic liposomes, metallic nanoparticles, solid lipid nanoparticles, and polymer-based nanoparticles, are used in CNS drug delivery. So here there are a few examples of the drug delivery methods. Like first one is with the help of liposomes. Next is magnetic and magnetoelectrical nanoparticles, and third one is polymeric through polymeric nanoparticles. There are some challenges that are associated with drug delivery system without the nanoparticles, that are like poor solubility, low bioavailability, reduced half-life, lack of selectivity, poor cell interactions, large size, difficult to administer. And with the help of nanoparticles, we can overcome all these challenges and ensure a good drug delivery mechanisms. Thank you.
Now, I would like to invite Yashika Garada. Yashika,
Yes, sir.
okay, start with your presentation.
Sir, is my screen visible?
Yes, very much visible.
Okay, sir. Thank you. Good afternoon, everyone. Myself, Yashika Ghara, a research student from Department of Zoology, Institute of Science, Nagpur, and I'm here to deliver a presentation on an immunohistochemical study of a gut neuroendocrine system in teleused fish, Trichopodes trichopterus pallas 1717. Starting with the introduction, the gut neuroendocrine system is the largest endocrine system constituted by several types of endocrine cells responsible for many functions, including digestion. Those enteroendocrine cells secrete various appetite-regulating neuropeptides like NPY, CART, leptin, gastrin, CCK, SCGN, as previously reported in teleused with the importance in control of appetite, energy balance, regulation of feeding, and digestion. Recently, the effects of prolonged starvation is extensively being studied in various other teleos like Tinca tinca, Cyprinus carpio, zebrafish, etc. And so, the present study describes the anatomical and histological features of the intestine and the effects of the short-term starvation on the distribution of NPY and SCGN endocrine cells in the foregut of the teleused fish, Blue-Gourami Trichopodes trichopterus. Moving towards the material and methods, the experimental animal of the present study is Trichopodes trichopterus, belonging to the Osphronemidae family of order Anabantiformes, is a hardy fish that abides variation in water quality parameters such as hardness, pH temperature, dissolved oxygen conditions, and also the visual transparency of the embryos of Blue-Gourami allows us a critical observation throughout the development and efficacy of mosquito control, making this blue gourami a great model to study. For the morphology and histology of gut, 6 adult gouramis of either sex were anesthetized and euthanized, and intestine were removed and fixed in bones for 24 hours. After tissue processing, the samples were proceeded for paraffin sectioning, and 8-micron thin transverse sections were stained with pass histological technique. To study the effects of food deprivation on neuroendocrinal cells of gut, the starvation experimentation was conducted. The adult gouramis were reared under natural photoperiod and were fed once a day with commercial food till the day of the experimentation. The fishes were grouped in 5 sets, each group containing the 6 number of fishes. The group 1, that is the control group, were continue receiving the food uh, on the day throughout the experimentation, once a day throughout the experimentation. While group 2 were starved for 24 hours, group 3 for 48 hours, group 4 for 74 hours, 72 hours, and group 5 for 7 days, and the gut samples were collected. The samples of foregut from each experimental group were dissected out, and the frozen sections were cut at 12 micron in transverse plane and were incubated in polyclonal antibodies listed in the table here, the neuropeptide by NPY and the secretagogue SCGN. And the immunoreaction was detected with the 3-amino-9-ethyl carbazole used as a chromager. Heading towards the result and discussion. Here, the figure A shows an image of an adult Trichopodes trichopterus showing the relationship of the intestine with the other organs in the abdominal cavity, and the figure B shows the gross morphology of the intestinal tract showing stomach, anterior intestine, middle intestine, posterior intestine, and the rectum. These are the photomicrographs of the transverse sections of the anterior intestine stained with pus, showing in inner circular muscle layer, the outer longitudinal muscle layer, epithelium, lamina propria, and the brush border. These are the photomicrographs of the immunohistochemical distribution of neuropeptide Y in anterior intestine of control group, 24 hours, 48 hours, 72 hours, and 7 days of starvation in epithelial lining of apical and middle region of lamina propria, having sac-like, tadpole-like, and elongated endocrine cells. And in the control negative, that is in the figure B, the control procedures by replacing NPY with the BSA resulted in the loss of immunoreaction. Similarly, these are the photomicrographs of the immunohistochemical distribution of the secretagoguin SCGN in anterior intestine of control group, that is figure A, and in 24 hours, figure C, 48 hours, figure D, 72 hours, figure E, and the 7 days, figure F of starvation in epithelial lining of apical and middle region of lamina propria, having sac-like, tadpole-like, and elongated endocrine cells, and in control negative, that is in the figure B, where the excision was replaced by BSA and resulted in the loss of immunoreaction. Here, in the table, relative density of the immunoreactivity of neuropeptide Y and the secretagoguin is indicated as highest, moderate, and the weak. From the previous photomicrographs, we can see that the immunoreactivity of the neuropeptide Y had increased with the starvation and found highest at the 48 hours of starvation. While in case of secretagoguin, that is SCGN, the immunoreaction was significantly decreased with the starvation and was lowest at 48 hours of starvation. Ending with the conclusion that the morphology and the internal structure of the intestine is similar. Intestine of the Trichopodes trichopterus is similar to other teleost, and even the short-term food deprivation. In the short-term food deprivation, the changes in the intestinal epithelium is seen. To our understanding, this is the first report to describe the morphology, histology, and distribution, as well as effects of starvation on NPY and SCGN immunoreactivity in gut of glucuramine. The result of present study suggests the need of investigation of histological and neuroendocrinal changes during long, particularly at the moment when food is no more possible. Those are the references. Thank you.
Now, I would like to invite Mittal. Mittal, are you here? Juhi Rani, Mukesh, Suresh, Daksh, Sunaina, Shatakshi, Chahat, Amit, Nikita, any one of you have left their presentation?
Yes, sir. I am left with the presentation. Nikita here.
Okay, start with your presentation.
My screen is visible, sir.
Yes, very much visible.
Good afternoon, all of you. I am Nikita Tushir from the Department of Biosciences and Technology from MMDU University. I am presenting my oral presentation on the topic targeted epigenetic editing of DNA methylation using CRISPR-Cas for stress tolerance. Starting with the intro, epigenetic modification to genomic DNA and histone protein shows increasing role in biological process. Due to the fact that epigenetic alteration do not change DNA sequence but instead modifies the DNA sequence to control their expression level. They only changes expression. They don't affect on the sequence like ATGC on that sequence. Third, in order to control gene expression, epigenetic uses a variety of mechanisms, the main ones being DNA methylation, histone modification, and non-coding RNAs. CRISPR-Cas genome editing is currently a mature biotechnological technology for enhancing crop growth and development as well as response to different environmental stress. Now coming to the mechanism of working of CRISPR-Cas system. And in this, as you can see the picture, there are two nuclease domain RUV-C and HNH and a PAM interacting domain. These domain clamps the targeted double-stranded DNA, which is and they creates a double strand break that is known as DSP. And there are two different methods of the gene replacement. First is gene knock-in or gene knockout. Nowadays, gene knock-in and gene knockout are the most popular techniques which are used for making transgenic plants using CRISPR-Cas. Coming to the next slide, CRISPR-Cas mediated genome editing on crop improvement. Control gene expression epigenetic uses a variety of mechanisms, and the main ones being DNA methylation, histone modification, and non-coding RNAs. CRISPR-Cas genome editing is currently a mature biotechnological technique for crop enhancing growth and development, and to the response to different environmental stress. And these stress are like abiotic stress, biotic stress, and yield of plants, etc. Coming to the application of CRISPR-Cas genome editing in crop improvement. Although Although it targets a specific DNA sequence, CRISPR-Cas can be used to add or modify expression of a specific gene that regulates plant growth and development, as well as its tolerance to biotic and abiotic stress. Now coming to the biotic stress such as drought, salinity, temperature and heavy metal present in a significant barrier to crop production and significantly lowers yield. Here are some examples of abiotic stress, plant Arabidopsis and rice. Uh, in Arabidopsis, we have a targeted gene that is shown in the slide and gene name, which helps in overcoming the drought stress in Arabidopsis plant. Likewise, another example, rice, we have targeted gene and gene name, which helps in overcoming the drought in rice plant. Now, next stress is biotic stress. 20 to 40% of the losses in worldwide agriculture productivity are attributable to biotic stress such as fungal, viral, and bacterial diseases. I have shown two examples of this also, tomato and rice. In tomato, fungus creates powdery mildew, and mildew and the targeted gene name is shown in the slide. And second one is rice. Fungus creates rice blast in rice, and if we create transgenic plants with CRISPR-Cas, we can avoid these fungus and bacterial or viral diseases in the plants. Thank you. My presentation is completed.
Anyone here who left with their presentation? Okay. After this, we will start valedictory session at 3 p.m. So join at 3 p.m. everyone.